Everyone, today we are going to discuss how to analyze MiSeq output data. And here is the file. Here you can see that how it looks like. So this data is coming as an output uh, from the after MiSeq analysis from the deep amplicon sequencing run. In this file, you can see the first column is showing as taxon level, and then rank ID with your speciation of data, and then taxon level of your species. So it depends on what are the your experiment. For me, I am working on Fasciola hepatica, gigantica, and Calicofendovini. So speciation is showing like that. So once you are, if you are doing with your microbiome, it will looks like differently with as per output of your data. And then you can see there is a daughter level that how your species daughter level are look like daughter level 3 or 1 or 0 like that. And then there are total number of sequence reads, and then in next columns there are samples which you run during your MiSeq analysis in the Illumina MiSeq machine. And these are different samples that you have run, and these are my samples which you have shown here, which I will explain in the next slide. So how we can calculate biases uh, which are produced during the deep amplicon sequencing or MiSeq run. So today presentation purpose is to remove biases from these sequence reads. Here you can see that these are different taxonomical levels are produced, and these are different number of sequencing reads that are produced against them. So for calculation of biases, we just need to select it all, or just with Control A, and then copy and paste on a new sheet, on a new sheet. And I did it earlier, and then I did some manipulation that I have selected only the required species like I am working on Fasciola gigantica, hepatica, and Coelicophendovini, and the rest of the species are removed, and I only kept these three rows which indicate all these three species on which I am working. So on the first row, you can see that these are different number of cycles that I have performed in the form of different samples. We are doing the deep amplicon sequencing biases removal experiment. So for this experiment, we did it like with PCR with 25 cycles, 30 cycles, and 35 cycles in each in triplicate. Uh, the eggs, this experiment was based on mock mixing of different fluke eggs. So each mix was in triplicate, and it was further run in triplicate. So the data is showing likewise. Here you can see the first uh, samples are from the 25 cycles, and then we have taken its average of the first triplicate, and likewise in the second triplicate we took the average, and in third triplicate we took average, and then finally average of all 25 cycles in the final column. And likewise we did with the 30 cycle and 35 cycle run. So in this way, first of all, we need to sort it out our data in a regular columns, so that we can make its further analysis with ease. And then I have made it this all copied and pasted in column wise. Here you can see, and then further uh, sorting out this data in the form of average values. We have now taken only the average values of each 35x, 30x and 30, sorry, 25, 30x and 35x average reads. Here you can see in each column, and these are the observed sequence reads that we got from the MiSeq run. And in the next column, we have taken the overall averages of reads from all three species. And finally, if we go further down, we have actual number of eggs that we have mixed in initially during the experimentation, which were 250 eggs from each species for each cycle of PCR, and then the total number of the sum of all reads, so that we can calculate the percentages. Then the further step is we will calculate the percentages of the observed reads. Here you can see from by using a formula showing over here that we have just divided, for example, the number of reads observed for Fasciola gigantica by total number of reads, and likewise for the others all. So here you can see that all average percentages are calculated for the actual observed sequence reads, and similarly with the percentages from the calculated from the averages of all. And further, if we go down, you can see the actual percentages that we mixed for each of the fluke species in the initial experiment are also calculated here. And then from this data, we can calculate the correction factor. The correction factor is calculated like we can divide the actual percentage over observed percentages. So in this way, we can calculate the correction factor. And after calculating the correction factor, now we are normalizing our data that we have observed initially. And in normalizing data, we have just multiplied the correction factor with the actual number of reads. Here you can see the actual number of reads. These are actual number of reads. These are multiplied with the correction factors. Here we have written the correction factor as we have calculated by dividing the actual percentages over the observed percentages of the reads. So after multiplying with this correction factor, we got the number of reads which were biased free. Here you can see here that. And similarly, we have generated biased free reads after multiplying with the correction factor with the average reads. So after getting this corrected data from the samples, we just paste it over a new sheet. Here in the first column, you can see the number of cycles, and further you can see that O is representing the observed reads, A is representing the actual reads, and C representing the corrected reads. So we will generate its percentage bar chart in the R, which I will show you after this slide. And similarly with the 30x and 35x, and with the average of above all like 25, 30, and 35x of PCR data. And these are the number of reads that we have calculated in the previous sheet: observed, actual, and corrected for all PCR reactions. And from this data, we will generate a percentage chart, which I'm going to show you just now. So here I will show you how we can produce percentage bar chart using R. Here is the R code that I have generated already, just to show you out that we are using tidyverse and readxl, two libraries for generating bar chart diagram. So first of all, we will give input data to the R. It will read the sheet 2 where we have put all our finally identified data, and then we will use melt data with ggplot and data long command, which will read all the data, and then it will calculate the percentages for each of the over group and each of 4 variables that we have put it into the Excel file. And then this is the important point here. I have made it X ordering. On the X axis, I have generated an order that first of all, it will show you the 25X bars, the observed with observed reads, then with 20x with the actual read, and then with the corrected reads, and likewise with the other number of cycles of PCR. So we can make ordering of x-axis. So if go further down, then we give it species order. We have 3 species: Fasciola gigantica, Hepatica, and Calica fendovini, and then we will use ggplot options to generate its percentage chart. And further, if we go down, we give a title to the plot, a proper title, and then x-axis and y-axis labeling, and then we have given the how we can make its grid lines and background and other things using themes. Different type of themes are given, and finally the plot will be generated and it will be saved in 600 dpi in png, and we can just run it here, go up and just give a run. It will run all the oh, the sheet is running. We need to close the sheet first, and then we can rerun it because it will only run once sheet is closed. Now you can see here that the it is producing a chart for us, and here you can see a clear bar chart with different percentage of mixes inside. Here you can see that the on x-axis you can see the number of PCR cycles as we have defined in the above section, that, and on y-axis the percentage of sequence reads, and these are different color combinations that we have defined in the script, and this is how it looks like. A beautiful chart is produced, and I hope you found this tutorial easy and in simple words. I made it for all of you, and if you like our videos, then please like, subscribe, and share of our channel with others. Thank you very much.